Hey everyone, welcome to our webinar. My name is Misha Proctor and I'm the Director of Marketing Communications here at Dialogue. I will be your moderator today. Our webinar today is titled Protect Your Anaerobes, Put a Whole Workflow Inside Your Chamber. First, a few housekeeping items. The webinar is recorded and we will be sending the link to everyone who registered, so don't worry. Also, if you have any questions during the webinar, please submit them through the Q&A chat feature. Some of you already submitted the questions, so thank you for that. We will be getting to those questions either during the presentation or at the end. Now let me introduce our speaker. Max Gravener is our product manager here at Biolog for instruments and software. He received his PhD from University of Georgia in microbiology with a focus on fungal genetics, where he worked with Candida albicans, studying natural variations among clinical isolates and how gene expression and regulatory differences can impact their ability to form biofilms on implanted medical devices. Welcome, Max. Please take it away.
Thank you, Kasha. As she said, my name is Max Cravener, and I'm the product manager for instruments and software here at Biolog. And today I would like to talk to you about how you can protect your anaerobes by bringing your entire workflow into an anaerobic chamber. Having the proper environment really makes all the difference when it comes to growing things. All sorts of variables need to be within a specific range, whether that's making sure that you're choosing the right plants to grow in your agricultural zone or planning to work and live on the moon. In the case of anaerobic bacteria, atmospheric control is really essential, as even the smallest amount of oxygen exposure, whether that's in just a short time or in minute concentration, can have a massive impact on the viability of samples and the reliability of your data. Oxygen is the enemy of anaerobic microbial culture. It produces toxic compounds in the form of peroxides and oxygen radicals, which can cause severe damage to cells and media alike. Facultative and aerotolerant anaerobes may be able to persist in the presence of some oxygen through the use of enzymes like superoxide dismutase, catalase, and peroxidase, or producing antioxidant compounds like uric acid and vitamins C and E. However, obligate anaerobes lack these key enzymes and the ability to produce these antioxidants, which makes them particularly vulnerable to oxygen exposure. Oxygen exposure can kill your workflow in many ways. Oxygen kills your time by necessitating repeats of experiments due to exposure causing failures. It kills confidence, as you may be able to see a strain of interest with preliminary sequencing data. However, you fail to isolate it due to oxygen exposure. Oxygen kills your credibility by causing growth inconsistencies and reproducibility issues among donor samples and synthetic communities. And it kills budgets by wasting time and high-value samples, draining your financial resources. And finally, oxygen kills media, as it's the leading cause of media deterioration, meaning shorter shelf lives and increased operating costs. This is why here at Biolog, we manufacture our anaerobic culture and transport media using our TruePress method, where the entire manufacturing process is done anaerobically, but more on that later. When culturing anaerobes, even a short exposure to oxygen while handling cells can cause instability in growth and therefore undermine your data reliability. So let's take a look at a couple of real examples of this. Here we have Bacteroides theta iota omicron, which was inoculated and incubated anaerobically inside a chamber. This plate has all the wells identical except for A1, that's our negative control, and you can see that the growth is very consistent across the entire plate. Here we have the same strain inoculated on the same plate, only this time it was brought out into the air during the inoculation step for just a few minutes. And the reason we did this was to modulate a situation where maybe you don't have access to a chamber, and so you're incubating in a box with an oxygen absorber. This small exposure when the box was opened, for instance, led to a significant growth variation across the plate, where all the wells should be identical, just like on the last slide. Here's another example, this time with C. diff, an obligate anaerobe, which grew very well inoculated and incubated inside the anaerobic chamber. However, just a few minutes of oxygen exposure during the inoculation step caused an incredibly significant drop in viability. These unstable results have the potential to confound drug sensitivity results or metabolism studies, leading to inaccurate conclusions. So how do we mitigate oxygen exposure during culture steps? Enter the AS500 anaerobic chamber, which presents a best-in-class solution for modular and ergonomic anaerobic culture with automated maintenance to make your life easier. The AS500 chamber is the ideal solution for anaerobic culture and handling for several reasons. First is the automated environmental control system. There's no need for daily baking of a catalyst as it's constantly heated to 100 degrees C, meaning that sulfides produced by microbes never have a chance to build up on the catalyst and therefore can't prevent the catalyzation of the reaction between hydrogen and oxygen. Further, protecting the catalyst is the Anaerobes pellets, which are activated carbon and potassium permanganate pellets, which filter out sulfides and other potentially harmful volatile compounds. Also included in the environmental control system is a HEPA filter, which helps to keep down particulates and spore contamination within the chamber. The AS500 chamber also includes a vacuumless pass box with internal and external touchscreen controls. This means that there's no need to cool your media or disassemble squirt bottles to prevent boiling and leaks, which can be commonly caused by vacuum cycles. The chamber also has pass-throughs for HDMI, USB, and Ethernet, which enables you to connect lab equipment and computers from outside the chamber to the inside. You can also add on a touchscreen monitor mounted to the incubator door so you can further integrate and manage sample information from within the chamber. Finally, the chamber is equipped with our vacuumless InstaSleeve entry, which means that you have immediate access to the chamber without having to waste time waiting for a vacuum cycle to complete. Conveniently, you can also bring in small items and materials through the sleeves, which further saves more time and gas and therefore your money. One of the key features of the AS500 chamber, and one of the ones that we're going to be talking about quite a lot today, is its modularity, where you can add up to 2 expansion modules to the main chamber, increasing the available workspace and increasing the number of users to up to 3 per chamber. While adding additional expansions for more workspace and users is helpful, sometimes this space just isn't enough. One of the biggest hurdles for anaerobic chambers is space. It's critical to have all of the needed materials in the chamber so that there are always things that are pre-reduced, ready to go when you need them. This presents an issue, however, when it comes to bringing equipment into the chamber. Space is always at a premium with anaerobic chambers, and sacrifices must be made, which means that anaerobes need to be subjected to sojourns out into the air to be handled in large equipment before being brought back into the chamber, which can jeopardize viability and reliability of your data. To address these space issues for equipment, we are now introducing the AS500 instrument module, the solution for containing large instruments and equipment in a fully anaerobic work environment, thus preventing the need to bring samples out into the air at all. How is the instrument module different from the existing expansion module? The instrument module is deeper and taller than the expansion module, accommodating instruments of up to 36 inches wide by 39 inches deep and 39 inches tall. Due to the significant depth of the chamber, reach does become an issue, so we added a side arm port and viewing window, so you always have access to your instrumentation, especially those power connections. We also have 2 options for the front panel, either the flat front or IM or a specialized front panel, which has been designed to allow the Odin 8 door or other instrument doors to open fully, called the IMO. A symmetrical build means the instrument module can be mounted either to the left or right side of either an existing AS500 or expansion module or another instrument module. But I'll talk more about all the available configurations later on. This chamber was based on a lot of feedback from customers just like you, all saying the exact same thing. We need more space. Labs in different fields have specific needs for their increasingly complex workflows, which require various pieces of equipment that might be too large to fit in most anaerobic chambers, like the AS500 or expansion module. For example, high-throughput processing of microbiome samples from donors often requires the use of colony pickers and liquid handlers due to the high volume of the samples being processed. Meanwhile, other research groups may need imaging and characterization equipment like the Odin-8, which when brought into the anaerobic chamber will give more reliable and more actionable data. Together with the AS500 chamber and the expansion module, the instrument module can create a fully anaerobic workflow so that your high value and sensitive samples are completely protected at all times from beginning to end, meaning more reliable data and more confidence in your results. So you've heard me talk quite a lot already about how an anaerobic workflow can really help. So now I'd like to move on to some real-life examples of how bringing your workflow into the chamber can significantly improve outcomes, especially for obligate anaerobes. So first, I'd like to go over some background on Odin and characterization of microbes with biolog tools. So Odin-8 enables the simultaneous incubation and kinetic reading of up to 8 microplates. And this allows you to monitor growth curves in real time and perform high-level analysis of kinetics, calculating parameters like area under the curve and growth rates, and finally compare samples to draw actionable conclusions. Our lines of phenotypic characterization plates make high throughput screening of communities and candidates very easy, allowing you to spot key differences in substrate utilization and sensitivities under a variety of different conditions. The characterization platform I'd like to focus on today is our phenotype microarray plate series, or PM plates, which screens samples for substrate utilization of carbon, nitrogen, phosphorus, and sulfur compounds, as well as sensitivities to pH, salt, and a variety of drugs and inhibitors, where each well of the microplate is a unique condition. In particular, PM1 and PM2 test the utilization of 190 different carbon sources, which probe a variety of pathways where growth can be measured over time. Historically, anaerobes were characterized by inoculating a PM plate inside an anaerobic chamber, sealing that plate, and then bringing it out into the air to incubate and be read in Odin. So here's an example of the type of data you can see with these plates. So first is a comparison of replicates for quality control and also to determine the stability of your data. You can see here in this wild type and mutant that they varied in their susceptibility to some of these drugs here in dilution series in wells A5 through 8 and D5 through 8, with pretty good reproducibility between the replicates. Then if we want to aggregate these replicates and calculate some parameters, we can do that as well to get some quantitative data. So here we have our wild type and mutant with replicates aggregated, and you can see the differences here between the mutant in blue and the wild type in pink. The mutant is much more resistant to the drug in this plate. And the values that you see displayed are the difference in area under the curve between the wild type and mutant. So the question that I need to answer for you now is, do you really need to screen in a truly anaerobic environment? Well, plate seals aren't perfect and they leak oxygen over time, which might be okay for facultative and aerotolerant anaerobes. However, with obligates, this is an especially critical consideration, which is why we developed the instrument module and made it so you can bring instruments like Odin-8 inside the anaerobic work environment. To demonstrate this, I'd like to show you some data that we generated using PM1 and 2, the carbon source utilization plates, both with a sealed plate method where it was inoculated in the chamber and brought out, and then with Odin inside the instrument module. First, we inoculated Bacteroides fragilis and obligate anaerobe onto PM1 and 2, and then sealed the plates in the chamber and brought them out into the air to incubate and be read in Odin for 48 hours. With this method, we found that there were 16 carbon sources which elicited growth by B fragilis, listed over here on the right. And notably, however, D xylose, shown in PM1 well B8, had some inconsistency between replicate 1 in black and replicate 2 in orange, where over the course of the 48-hour assay, replicate 2 actually picked up growth several hours earlier than replicate 1. So repeating the same exact experiment, but this time bringing Odin-8 into the anaerobic work environment instead of sealing the plate, we found an additional 7 carbon sources for a total of 23, which were able to elicit growth in B fragilis, again listed over here on the right. And taking a look at D xylose again in well B8 of PM1, we can see that the replicates now overlap perfectly, demonstrating their reproducibility is better when the whole workflow is brought into the anaerobic work environment. So taking a look at these data side by side, we can see that this really shows just how much can be missed when screening obligate anaerobes using methods which might expose them to even the smallest amounts of oxygen, like the plate seal. Without Odin-8 in the anaerobic work environment using the instrument module, we would have missed out on potentially valuable data, including utilization of fucose, lactulose, dextrin, N-acetyl neurominic acid, this arabinose compound, beta-methyl D-galactoside, and finally stachyose. I would also like to emphasize that several of these substrates, like lactulose and beta-methyl D-galactoside, likely took longer for B. fragilis to change over to process, which may have been inhibited by O2 leaching in through the plate seal in the second day of incubation. But we were able to capture these substrates by incubating and reading inside of the anaerobic chamber. So I hope that these experiments have convinced you that bringing your workflow completely inside the anaerobic work environment can have significant benefits to your data quality and its reproducibility. So next, I would like to talk a little bit more about the available configurations for the anaerobic chambers that we have, as well as the types of instruments that we know can be accommodated inside the instrument module. Your workflows can be very complex, and we know that, which is why we've strived to create a flexible platform for anaerobic culture and specimen handling through our modular anaerobic chamber system. Whether you're using colony pickers and liquid handlers to process hundreds to thousands of high-value donor samples, or you may need an imaging system or characterization equipment like Odin, or you might need a refrigeration unit for low-temperature applications or media storage. We have you covered regardless of the application. Our flexible anaerobic chamber configurations mean that we can make a custom-tailored chamber designed for your specific workflow needs. When it comes to the available configurations for the AS500 chamber, we have a few general guidelines. The AS500 chamber can be purchased in both the standard right side passbox or a left side passbox version. These two chambers can also be attached to each other for a continuous workspace with 2 incubators and 2 passboxes. For the modules, the general rule is that up to 2 modules, either 2 expansion modules or 2 instrument modules, or one of each, can be attached to each chamber. This brings us to our maximum configuration size of 4 modules mounted between 2 chambers. For our modular configurations, we also have options for pre-assembled desk height, or 29-inch, and counter height, or 36-inch benches, where the chamber's modules ship mounted to the bench to be joined together on installation. That way we can accommodate just the right ergonomic setup for your needs. We can also ship standalone chambers pre-assembled with a hydraulic bench for optimal ergonomic adjustment, or either of those fixed bench options. When you purchase an anaerobic chamber from us, the installation and training will be performed by a biolog technician or one of our authorized distributors. And we also offer annual preventative maintenance and extended warranty packages. The chambers also hold a number of certifications, including FDA and MDEL, as well as CE and EN-ISO. We also offer the only hands-on chamber and anaerobic bacteriology course out there, hosted at our Morgan Hill, California location. This course is broken up into 2 portions. In the classroom, you'll learn all about oxygen toxicity, specimen collection and transport, as well as how to select just the right culture media for growing, isolating, and identification of your unknown anaerobes, as well as proper handling of microbiome samples. That way you minimize any disturbance to the community structure during handling. In the lab portion, you'll get extensive hands-on experience with working in the chambers in real-world scenarios, focusing on isolating and identifying anaerobes using determinative culture media. Finally, you'll have ample opportunities to learn more about the operation and maintenance of the anaerobic chambers, as well as detailed troubleshooting information, just in case. I also wanted to mention that Biolog Lab Services is now able to perform anaerobic testing in addition to our existing aerobic services. So if you're not able to get a chamber, or if you just want to test things out, please do not hesitate to reach out to us for all of your anaerobic needs. Biolog's pillars of microbial mastery include our three major lines of products and services, which you can leverage to fully understand your microbes. First off, we have our phenotypic analysis and identification pillar, defined by the Odin family of instruments, Odin L, Odin 8, and ID Station, powered by our analysis and identification software platform, allowing you to characterize and identify thousands of bacteria and fungi, as well as characterizing mammalian cells if that's your thing. As you've heard already, we also offer anaerobic sample prep and handling through our anaerobic chambers in the AS500, which you've heard about already today, as well as our compact model, the AS150. We also have our TruePress culture media and transport media, both of which are manufactured under totally anaerobic conditions from start to finish in a unique process to prevent any toxic oxygen radicals from forming in the media, leading to more reliable results. Finally, our certified lab services pillar includes genomic, proteomic, and phenotypic identification services, as well as services like enumeration and phenotypic characterization and screening with Odin, both for aerobic and anaerobic conditions. That about wraps things up, so thank you all very much for tuning in. I hope that today's presentation was insightful and helped you understand how bringing your entire workflow into the anaerobic work environment can help to save you time, money, and effort while increasing reproducibility and confidence in your results by protecting your precious anaerobes. We will start by taking some questions from the chat, but I also wanted to ask that you just take a couple minutes after the webinar to answer a few questions in our survey. Thank you all again very much, and please do not hesitate to reach out for more information at info@biolog.com. Now we'll move on to our Q&A session.